Right. Welcome, everyone. Thanks for coming back this morning. We're so grateful that you're spending your weekend with us. So I want to talk about the stages of drug development and some lingo that you might hear when you read about clinical trials, when you're you're maybe doing a search on the internet for what's happening, and there's a kind of a vocabulary. And we're all now in this exciting and unprecedented time for CMT where there's a lot of clinical trials ongoing, so you might even be thinking about being part of a clinical trial. And so that's really how this morning is going to evolve. I'm just the warm-up act, really, for the talks that come later this morning, but we're going to be talking about what are these stages and what it might feel like to be part of a clinical trial. So you might think that drugs get marketed or approved for marketing because they work, but it turns out that's not exactly true. The reality is drugs get approved for people because they have a relative balance between benefit and risk that's positive for the people who are going to use it. And all three of those pieces are really important for how we think about how drugs work and how we might market drugs. So the benefit, the risk, and the people who are going to use it. And you always want to be maximizing the benefit and minimizing the risk, but it's a trade-off. And we try to understand what that trade-off is through the process of drug development. So how do we know if benefits outweigh risks, and how do we measure benefit? So benefit is the part that feels like, how is it working? Outcome measures, Dr. Burns is going to talk about this next, but something that tells if it's making a difference for you as a patient. Typically, we'll do this by comparing two groups. We'll compare a group of patients who are on treatment against somebody who's not on treatment. So they might be getting a placebo, like a sugar pill, or they could be a natural history, but you're looking for a difference between those two groups. So in one scenario, you get the treatment and you get better, and the placebo group stays the same or gets worse. But it's also possible to have a positive benefit risk if the patient who gets the treatment stops progressing. So as we know, CMT is a progressive disease. So stopping that progression when people who are getting that sugar pill continue to get worse, that's still a win. That's still a positive trial. And you'd see a statistical difference between those two. For a disease that there's no approved treatment, what that benefit looks like, we're guessing a little bit. We're still trying to figure that out. That's why we invest in clinical trial readiness. That's why we invest in outcome measures. But we're really trying to figure out how to best measure that in a slowly progressive disease. What we don't want to do is do a trial that takes 5 years or 10 years, because no patient wants to be in a 10-year trial, and I can assure you no drug company wants to run a 10-year trial. That's just too long. And so there is another pathway called the accelerated approval pathway. You might have heard about that, and that's just about finding some other way that's a little bit faster to get to your answer. So instead of waiting for that 5-year answer or that 10-year answer, we try to find an outcome that works maybe in a year or two, and that's called an accelerated approval. If you get that kind of approval, you still have to do a confirmatory study later, but you still get to the end point. So all of that is kind of in the midst of what we're figuring out in CMT drug development right now. So how do we measure risk? Risk is about things that go wrong, side effects. When you think about the side effects of drugs, how well is it tolerated? And so as you go up to more benefits, sometimes the risks change a little bit. And we talked about what is the group of patients in which that's going to be used. That's an important measure when we think about risk. You might feel different about risk if your disease is slowly progressing over 20 or 30 years than you feel about risk if you have a child who's going to be in a wheelchair in their teens. So you might be willing to tolerate a different level of benefit risk in that scenario versus another scenario. So all of these three pieces matter for each subtype, and they matter for each drug. And so we're trying to figure all of that out. In terms of the population, we talk about CMT as a rare disease, and sometimes you also hear this term orphan disease. These are used interchangeably. It turns out orphan disease actually has a definition and a legal and a kind of a regulatory setting. So the Orphan Drug Act, which was approved in 1983, wildly successful piece of legislation, opened up a pathway for rare disease that's accelerated drug development and really kind of spawned the whole biotech industry. It creates incentives for companies that are financial. It gives flexibility around regulatory pathways. It considers the feasibility of what you can really do in a small population, which is different from what you can do in a bigger population. And importantly, it gives this period of regulatory exclusivity. So for 7 years in the US, you get an orphan drug approval. There's not another drug that can come in for that's the same as yours for the same indication. And so that's really kind of opened up the doors to drug development for diseases like CMT and other rare diseases. And so we're always talking about why it's important to support rare disease drug development, because all of these things work together. So we're going to talk a little bit about that sort of lingo that I referred to. I mentioned that we measure risk and benefit. Risk we think of as safety. Sometimes benefit, you hear this word efficacy. Is the drug efficacious? That's kind of how it works over time. And we really think about risk and benefit at every single step of drug development. But in the very beginning, it's a little bit more focused on safety because you don't want to go too far if the drug isn't safe. And so there's a heavy weight on safety and risk at the beginning of drug development. And at the end of drug development, when you're feeling better about that, you start to ask more questions about efficacy. And so that changes over time. So before the drug goes into the clinic, into patients, we call that preclinical. In that preclinical phase, we're asking basic biology questions. These are things you do in a lab bench, things you do in a test tube, things you do in a petri dish, things you might do in a mouse in Jordan's lab. All of those things are happening to really test in ways that it's harder to test in people. Is this safe to move forward? Do we understand the mechanism? Do we understand the biology? And once you feel confident about all of that, you start clinical trials. So clinical trials, and you might hear these words, phase 1, phase 2, phase 3. So we'll talk a little bit about what those mean. So in a phase 1 study, your real question is it safe? In the most classic phase 1 study, you're really only looking at safety. So you might not even start in patients. You might just start in healthy volunteers. And you're not even asking any questions about efficacy or benefit. You're just saying, is this safe to use in people? And so these are people who are just willing to sign up for that kind of work. Sometimes you hear about phase 1A and phase 1B. You might hear a little bit of that this morning. So in a phase 1A trial, you're just trying a single dose and you're just going higher and higher and higher. And you're seeing how much can be tolerated by people. In a phase 1B trial, you take a few doses and then you just go higher and higher and higher. And then you stop when you get too many side effects and you say it can't be tolerated anymore. And that's a classic sort of phase 1 study. In phase 2, you're kind of getting ready for the big step, which is ultimately phase 3, but you're starting to ask more questions about efficacy. You're starting to ask more questions about benefit. You're using more of those outcome measures to kind of pave the way for the big, expensive trial that you're going to do to get your drug approved. Sometimes you're asking about questions like, what does this drug do to my body? But you're also asking, what does my body do to this drug? Sometimes called PK/PD, because our bodies metabolize drugs in different ways, and that can affect other parts of the body besides the parts you think about for CMT. Our liver, our kidney, all of those also have to be considered. And you start to go not just from phase 1 where you're saying, what's the highest dose I could possibly look at, but what's that benefit versus risk? What's that window in which I'm maximizing benefit and minimizing risk? And you try to get to kind of a smaller range and a better idea of what those outcome measures might be. Then you go for the big one, phase 3. We call that the pivotal trial. In the pivotal trial, this is where you really got to say, is this worthy of being approved? In a big indication, you have to do 2 big phase 3 trials. You might have 10,000 patients if you're studying something for blood pressure or diabetes. You're not going to have 10,000 patients for an orphan disease like ORC where you only have 1,000 patients. So you're always kind of figuring out what that looks like. These pivotal studies are the ones that really got you approved. And the trick about it is you have to pick the winner. You got to choose an endpoint, and you got to say, I'm going to base my statistics on this one endpoint. And remember now, for CMT, we don't have anything approved. So we're still a little bit in that guessing phase. We're in that figuring it out phase. And so it's tough because there's no roadmap where you can say, I'm just going to do what everybody else did. I'm going to follow the other pathways. So the companies that are in clinical development right now are working together and with us and the whole community to figure out, like, how do we measure this in a way that makes us comfortable that the benefit versus risk is in the right place? And we're all figuring that out together. But you have to make that one choice. This is my primary measure. This is my primary outcome. If this works, it's great. You're golden. You get your approval. And then you take the drug onto the market. You get to sell it to whoever you want, people who have that indication. Use it, but they're still measuring benefit and risk in this population, and particularly from a safety perspective. So when you're testing in a small group of patients, you want them to be very representative of the group that will ultimately use it. But if you have something that happens rarely, a rare side effect, you might not see it until you go onto the market. And so these are called post-marketing studies, and companies continue to look and ask those questions. What is the balance between benefit and risk in that setting? And if you are accelerated approval that we talked about a little bit earlier, so maybe you used a biomarker or some other way to get to the finish line that takes a little bit less time, this post-marketing will actually be a confirmatory study. It'll be a clinical study where patients enroll very much like phase 3. So that's another important part and another thing to consider. So in classic drug development, you do a 1A, 1B, a 2A, 2B, 2 phase 3s. But in orphan, that can be really hard to do. It's not always feasible. And sometimes you hear phase 1, 2, phase 2, 3. And I think in that situation, it's really about just combining the phases. So instead of starting in a healthy volunteer, you might go right into patients and start asking some questions about dose. And then you go to one more trial, and you're really going straight for efficacy. Because if you only have 1,000 patients in the US on 4C, for example, you don't have to put all the patients in the whole world in your trial. You're looking for a small group that's going to be representative. It is a little bit tricky and something that companies have to think about. What is that group in your clinical trial, and how well do they represent the population of patients who are going to use the drug ultimately? And one of the tricky things, and you might see this in your own family, is not everybody progresses at the same rate. So there is, we call it heterogeneity, right? People are all just a little bit different. And so you're trying to measure change in this group that might have a whole range of difference and sort of a bouncy baseline, and you're trying to find a difference. And so that's the other trade-off that drug companies are trying to make. And Dustin said earlier, 90% of drugs fail. This is the part where it gets tricky. Like, what does that group look like? Can we let everybody in the trial? And the answer is no. You've got to think about, how does that group represent who will use it, but also what's a group that I can use to kind of fairly understand how this drug is going to work. And so then you have what we call inclusion and exclusion criteria, who can be in and who can't be in. And that's really the company trying to figure out how to get a group that's not too variable, that's going to be a fair representation, but still a group that's going to help you answer that question. How does it work? And so we see all of that as part of the decision about how do drugs get developed and who gets to get in trials and who doesn't get into trials. And so I think as we see more and more drugs coming through the clinical pipeline, I think we'll feel that as well in how those decisions are made. And that's something that the drug developers will talk about a little while. So why do trials fail? It's important to know that a failed trial doesn't always mean that the drug didn't work. Sometimes things don't work because the benefit risk isn't right for that group. And again, it's that balance. How do you feel about the benefit versus the risk for that clinical subtype that you have? You might have got the wrong patient population. There might just be a little bit more variability in the group than you expected. So you think about, then you look at those two groups, and they separate, but they don't separate quite far enough to give you statistical significance, right? So they look different, but not quite different enough. Not uncommon in rare disease in particular, wrong outcomes, right? This is a tricky one because we're still figuring it out. What if I put all my money on, an endpoint that it looks pretty good, but it just didn't hit that statistical significance? It turns out that there is some flexibility out there, especially in the rare disease space. You can have a conversation with FDA and say, look, I didn't make this one, but I've got some other evidence. Let's have a conversation. They can actually often be really helpful and accommodating. And sometimes that happens. You just put all your money on one thing and it didn't work out because maybe you picked the wrong endpoint. Maybe FDA said to you, hey, we don't know anything about CMT either. We haven't created this roadmap. Something worked for muscular dystrophy, why don't you try that one? And you get stuck with it a little bit. So it's all a conversation. What really brings value to patients? What is that benefit? Have I measured it in the right way? Have I measured in a way that matters and is important, is clinically meaningful to patients? And are the risks worth it? Because every drug has benefits and risks, including the aspirin you take and the Advil you take, and not every drug works in every person in exactly the same way. And so as much as we measure risk-benefit for a population, for each individual person, it's also going to be different. And so you're going to have your own things happening in your body, and so everybody has to make their own decision for themselves about whether the benefits that you could see are going to be worth the risks that are always there. So how do we work at the CMTA to help drug development? And you've heard some of this already today. The foundation of everything we do is our strategy to accelerate research. This is what supports all of the R&D work. We invest from very early kind of bench test to kind of scientific studies, the preclinical toolbox and the preclinical phases, and we support studies all the way from phase 1, 2, 3. We're ready for phase 4 if it comes. We hope it's coming soon. And we just are in this unprecedented time with all of these clinical trials where it feels like after all these years of investing in outcomes and readiness, as well as the science, because you got to have both. You got to have the drug and you have to have a way to measure whether or not it's working. We're starting to feel some momentum. We're starting to feel some excitement. And I think one of the things that happens is more and more companies get involved, more and more companies get excited, investment comes in, momentum continues to build. And I think that's the point at which we're at right now. Our support of clinical trial readiness has been really important, funding natural history studies, outcome measures, and biomarkers. With over $33 million to date spent on research, we're so proud at the CMTA of all of the work that has come out of that and all of the great things that have come out of that. You heard Catherine speak earlier about the Inherited Neuropathy Consortium. We call it the INC for short. This is a group of 25 sort of world-leading experts, like the best of the best all over the world that everybody goes to for CMT advice and research because they've been in it for their whole lives. They've dedicated their careers to it. And they've formed this consortium to create a place where CMT research could be discussed and published and working together, and created a database for patients. And if you go to an INC site, you could join the INC patient research group, and it would be a part of this tool that is out there for scientists, for researchers, and ultimately drug companies to use to further basic research. So over that 15 years, in 8,500 patients worth of data has been collected. The NIH funds these consortiums for 15 years. They do 3 5-year grants, and then they say, hope you guys are sustainable. And so that funding ended at the end of last year. And so the CMTA stepped up and stepped in to support the INC. That's a big commitment for us. It's $1.2 million per year, but so fundamental to preserve that data and continue to grow that database, continue to build that database so that it's available to researchers and available to drug companies to accelerate research. And we're super proud of that partnership, and I think it gives us a really special place in the drug development landscape. And so finally, the last piece of it is the patients as partners in research. Catherine will talk about that later this morning, but this is people who sign up to be helpful and supportive and let us reach out to you so that you can play a role in drug development, which might include participating in a clinical trial like we talked about today. But it could just be giving advice. This thing about outcome measures, how do we understand what really makes a difference for patients? Turns out you ask patients. And so to help drug companies understand those kind of things has been really super powerful. So 2025 brings more clinical trials to the CMT community than ever before. We're really excited about this kind of pivotal year in the history of CMT, and it's so exciting to be here in this room with the CMT community. So the rest of the morning is going to be really exciting. We're on break until 11 o'clock when Dr. Burns will speak about outcome measures, and then we'll hear from the drug companies about how they're thinking about drug development. And so please be back here at 11. Enjoy a little bit of a break.